Alright, so let's talk a bit about how the mRNA vaccine for COVID-19 works. In a human cell, the first step of translation is known as initiation and occurs when mRNA travels from the nucleus through the cytoplasm to the ribosome. Ribosomes are composed of two subunits, and the smaller one is a subunit that binds to the beginning of the sequence, where a tRNA molecule carrying an amino acid called methionine binds to the start codon on the mRNA sequence, initiating translation. The region upstream of the start codon is referred to as either the leader sequence or the 5' untranslated region. Each tRNA molecule holds both an amino acid and an anticodon. The anticodon will bind to the mRNA sequence, and the complementary amino acid will be released from the tRNA molecule and bound to the polypeptide chain in the larger subunit. How does a tRNA know which amino acid to bind? This step is accomplished in what is referred to as tRNA charging, where an enzyme called aminoacyl synthetase binds to the 3' end of the tRNA molecule by its carboxyl group. Each amino acid has its own version of this enzyme, so specificity can be optimized. The elongation phase of translation is the stage in which the tRNA matches each corresponding amino acid to the codon sequence of mRNA and binds them into a polypeptide chain. This process will continue until all the codons are read. Once all the codons have been read, the tRNA will read a stop codon that will end the process and release the translation complex. It should also be noted that following the stop codon is the 3' untranslated region, which helps regulate genetic expression and can also influence the stability of the mRNA sequence. The mRNA vaccine uses nanoparticles to deliver mRNA to a human cell, which in turn undergoes translation and produces the protein the mRNA is coded for. In the case of the COVID vaccine, this protein would be the antigen for COVID-19 that helps the virus enter our cells to begin infection. Once the antigen is produced by our cells, it floats freely in our bodies until T lymphocytes recognize it as a particle that is not nascent and destroys it. This builds up immune memory to do the same with other particles resembling that spike protein. This new method for creating vaccines is more efficient than traditional viral attenuated vaccines because variants can be more easily created by simply changing the mRNA sequence to reflect antigens on any viral mutants. It also doesn't involve any infection of our cells like viral attenuated vaccines do. In the case of a non-synonymous mutation, the COVID-19 vaccine would likely still be effective against infection due to the fact that these types of mutations typically do not result in changes in protein structure. Synonymous mutations, however, do, and development of a new vaccine would need to take place in order to produce a new antigen for our immune memory bank, if you will. These types would involve alternating the mRNA sequence to produce a new vaccine with a new antigen.